Today we will be doing lecture 4, which is the membrane transport. It's a relatively short lesson compared to the other ones. So maybe we'll have some time to review for the quiz next week. When more people get here, I'll go through what's expected on the quiz, as well as I will talk about how to do the labs for next week. So let's get started. Membrane transport. Now, to understand membrane transport, we first talked about the various functions of membrane proteins. And then here's a chart that shows you the key functions associated with membrane proteins. But first, let's just quickly refresh your memory of what the cell membrane is made up of. Okay, so cell membrane. So in the cell membrane, the major component is the phospholipids, right? You remember the phospholipid is basically a type of lipids, and it's made up of glycerol plus 2 fatty acids, as well as a polar phosphate group. Okay, so that makes the molecule partly hydrophobic and partly hydrophilic. So your cell membrane is going to look something like this. Okay, and this would be like the outside of the cell, the outside, which is what we call the extracellular fluid. Okay, and then this would be the inside, which would be the cytoplasm. Okay, and then here, basically, if this is the cell, I'm zooming in this portion here. Okay, so it's 2 layers, right? We call this the phospholipid bilayer. Okay, so 1 layer and then another layer. Okay, so what we have is the hydrophilic head. Okay, hydrophilic means water loving. They will interact well with the liquid environment, mostly water, the ECF. Okay, and the cytoplasm is also mostly water. You would also have the hydrophilic head on the inside. And then sandwiched between the hydrophilic heads would be the tails. Right? Tails are made up of fatty acids, and so they will be hydrophobic. Hydrophobic tails. Now that's quite nice because it will create a barrier to prevent things from going in and out, without regulation. Now, every now and then you would have a protein, a membrane protein that get inserted. Okay, so if a protein goes all the way through, right, from the outside to the inside, we call this an integral protein. Integral protein goes all the way through. Okay, and then sometimes you might have protein just kind of stick to the side. Stick to the side like this. Okay, so these are called peripheral protein. Okay, and their function might vary depending on the type of protein that you find. But this classification, integral or peripheral, is not a category based on function. It's category based on how it is inserted in the membrane. If it goes all the way through, integral. If it's just attaching on either the outside or the inside, peripheral. Okay, now that we have an understanding of the basic parts of a cell membrane, we will talk about the functions. So the first function, as shown in this diagram, is the membrane proteins. They allow the attachment to cytoskeleton. Okay, remember cytoskeleton? We learned about it last week in the last lecture. There are different types of cytoskeleton. It's made up of things like microfilament, right, or actin filaments. It's made up of intermediate filaments as well as the microtubules. So in this picture, like these green things, these purple things, and these yellow things, those are the cytoskeleton. Okay, cytoskeleton. Cytoskeleton. Right? So something needs to be able to attach to it, right? And that's what the membrane proteins are for. So in the diagram here, maybe there are some cytoskeleton here on the inside. Okay, so this part would provide like an anchorage point to attach to the cytoskeleton. Same thing on the outside. So by attaching to the cytoskeleton, it allows the membrane protein allows the cell to maintain and support cell shape. Okay, by providing anchorage point. For things like actin, right, which makes up your microfilament, and intermediate filaments. To attach to. Okay, so that's the first function associated with membrane proteins. Okay, for those of you who just joined, I'll give you a moment to maybe quickly take a picture. I've created a folder on eCentennial that says study guide answer. So now you can find them easily. Maybe I'll just quickly show you where that is, so that you would know. Okay, so like if you go to content, and on the side here, right, lecture, where you usually find your recordings, there's a study guide answer. So if you click in it, then you have study guide 1, 2, 3, and then after class today, I'll put number 4 there, right? So you have it all in one place. It should help you with studying. Okay. Next function is cell signaling. Cell signaling. So a lot of hormones, well, we know hormones have two categories, right? They are either made up of lipids or they are made up of proteins. So they function a little bit differently. The lipid hormones, right, the steroid hormones, because they are steroid based, they would have no problem crossing the phospholipid bilayer. Okay, so they would go into the cell, and then they would go directly to the nucleus and cause changes in which gene gets turned on, which gene gets turned off. The protein hormones have a different pathway. They cannot cross the membrane easily. Okay, so the only thing they could do is they will remain outside and they would bind to a membrane protein, which will then cause a series of changes within the cell. Okay, so maybe I just give you a brief note here, right, to help you understand that. So hormones, right? There are two types. It can be steroid hormones. Okay, so these kind of hormones, what they do is this is the cell, this is the nucleus. Okay, so the steroid hormone, okay, maybe something like this. Okay, you know what, that's too difficult to draw. I'm just gonna do a blue dot. Okay, steroid hormone, they will go right through the cell membrane and directly into the nucleus, and then it will change gene expression. Okay, gene encodes for proteins, right? We talked about that. So it will turn on some gene, it will turn off some gene depending on what steroid hormone it is. But the point is, it's able to go in directly affect the nucleus. On the other hand, we have the protein hormones. Protein hormones. Okay, so these one cannot go into the cell. So if there's a cell here, this is the nucleus, you will have to have a receptor protein on the surface. Okay, this is a receptor. Okay, and then when the protein hormone comes, it will bind to the receptors, and then it will cause a series of reaction, right? Signal pathway to go on. Okay, so this is called a signal transduction cascade. It basically is like a fancy way of saying a bunch of reaction occurs. It's like broken telephone, okay? Except this time the message is not altered. Okay, so you pass it down, and then it will change gene expression in the nucleus. Okay, so the protein hormone does not physically enter the cell. It relies on the protein receptor. Steroid hormones can just go in directly on its own. Okay? So that's why we need to have a membrane protein on the surface to help relay the signal. Okay, so protein hormones cannot enter the cell directly. Okay. It binds to a protein receptor. Okay. Protein receptor, which will relay the signal and cause changes in the cell. Okay. So in the lecture, I talked about adrenaline being such an example. We talked about neurotransmitters, which is how neuron communicates with each other. Those are examples of how cell signaling is going to work. Any questions so far? Okay, another example is enzymatic activity. Enzymatic activity. So we know enzymes are proteins that catalyze chemical reaction. Okay. Many reactions in the body require multiple enzymes. Okay, so for example, if you start with material A, substrate, right? That's the word we learned. If you start with substrate A, it gets turned into B, which gets turned into C, which gets turned into D. Like this would be like the final product. Okay, final product, whatever it is, doesn't really matter. Maybe you make an amino acid, okay, in the body, okay, and this one is the starting material, okay, starting substrate, okay, over here. Okay, so A is the starting substrate, and this is the final product at the end, right? Just put them in bracket maybe. So it takes 3 steps, and each step you are going to need to have an enzyme, right? So here you would need like enzyme number 1, enzyme number 2, enzyme number 3. So sometimes it's easier if you put those enzymes close to each other, right? So that the product from the first reaction doesn't have to, diffuse around to find the second enzyme, right? It's just right next to it, next to the first one, and then you can catalyze the reaction much quicker. Okay, so this would be like enzyme number 1, this would be enzyme number 2, and then maybe we can add a third one here, right? That would be enzyme number 3. So the product of the first reaction will be used as the substrate of the next. And then this product of the second one will be used as a substrate of the third. And in this way, you were able to catalyze a reaction faster. Okay, so just an other point I would add here. Enzymes belonging to the same metabolic pathway are usually placed close to each other, right? So on the membrane in this case. Give you a moment to catch up. Okay, next, this one. This is intercellular junction. Intercellular junction. Okay. A junction is basically connecting two adjacent cells together. Okay, so membrane proteins of adjacent cells that are next to each other can hook on to each other. To form various types of cell junction. There are different categories. We didn't go into the details. Example, you will want to have this in the intestinal cells. Okay, you don't want the food to be leaking out, right, from your intestines. So you want a tight seal between the cells. Okay, so the junctions prevent uncontrolled movements of substances between cells. Okay. All right, next here, you know, we have two cells. This is one cell number one, and this is cell number two. Okay, same thing here, right? This is cell number one, cell number two. Okay, so another function is cell recognition. Okay, how does a cell know what belongs to the body and what does not? Okay, so there are many different molecules on the surface of a cell. Okay, these are called antigens. Okay, so what molecules are found on your cells are going to be different from what I have on my cells. It's going to be different from, in fact, everybody on this planet. Okay, unless you have a twin, then you know you're going to have the same kind of cell surface antigens. But you know, people will have similar antigens, but no two individuals will have the exact antigens, right? That's why, you know, if you think about organ transplant, you can't just take the liver from any random person and then, transplant it into someone else. Your body's going to reject it. And the principles of rejecting organs is basically the underlying mechanism is cell recognition. If your white cells, okay, so this cell number 2 is going to be like a white blood cell, for example. If the white blood cells detect a foreign antigen on a cell, then it will attack it. Okay, so foreign means outside of the body, right? If a cell has antigen that does not belong to your own body, okay, maybe it came from a bacteria, maybe it came from a virus, maybe it came from a transplant organ, right? Then your white cell is going to attack it. Okay, so this ability, this cell recognition, it is the key to how the immune system works. Immune system works. Okay, so your immune cells will be able to detect foreign antigen. I don't want you to think antigens are bad. Antigens are just stuff that are on your cells. And if there are foreign antigens, foreign stuff on a cell, it tells the body this cell is from the outside and it shouldn't be in the body. And that's how they attack it. Okay. Any question? No. Okay. Next is transport. Okay, transport. Because the phospholipid bilayer is primarily hydrophobic, so a lot of things will not be able to go through. Things that are too big will not be able to go through either. So many substances, okay, for example, large particles, charged, right, molecules rely on membrane proteins, right? Specifically integral proteins, right? We talked about that at the beginning. Integral proteins to move across the cell membrane. Okay. All right, so let's take a pause here, take a moment to stop, and I will talk about what's going on next week. Okay. So according to the calendar, which I am sure you are aware of, let's pull the calendar out. Okay, so you can see we are where are we today? We are the right here. Okay, we're October 1st. And so next week there will be no virtual tutorial. Okay, there will be no synchronous class. And there is no new lesson to learn next week. Okay? So what you want to do is you want to do your Beetroot lab, which is 10% of your final grade. So that's quite a bit, right? And you also want to study for your quiz number 2. Okay, so quiz number 2 is exactly like quiz 1, the format, right? There's going to be multiple choice, multi-select, and some application questions. I will post the detail after class. Okay, it will be on eCentennial and you can check it. And it will be available from 10 a.m. to 10 p.m. Just like last time. All right. And in addition, you want to be working on your Beetroot lab. So you might have already done that. And if you finish it, you can submit it. So for those of you who don't know where the Beetroot lab is and how to submit, I will quickly show you. So when you go on eCentennial, I will actually post an announcement after class as well. Okay. You know what? Maybe I will just post it right now, so you will have access to it right away. So let me go to the news tool. Oh, that is weird. I thought I typed it up already. but for some reason it did not save. That's okay. I will do that after class. Okay. Did it save for the other class? Let me see. Nope, it also did not save, which is unfortunate. I don't know what happened. Okay, so anyways, what you would do is you will go to content. Okay, content. Oh, I know why. Sorry. I'm student view. That's why it didn't show up. Okay. Sorry about the confusion. Let me just go back to here. Go to new news item. Now, go to news tool. All right, there is the week 4 stuff. Okay, so let me publish this, and then you will all see it. Published. Okay, there it is. Okay, so when you go to the course homepage, you will see, typical access of the tutorial recording, and then you can complete the lab 1 beetroot experiment. So you can just click on that. Okay, you can just click on that. Now let me go back to student view so I see what you will see. So you click on the beetroot lab, and you can read this stuff. Now we cannot physically do the lab, as you would understand why, for obvious reasons. So you will watch this YouTube video. Okay, this is someone else doing the lab. Okay, don't skip through it. Like just sit there and watch the whole thing. It's just like 8 minutes, and it's quite detailed. And take notes while you watch it. Okay, because some of the questions I ask you is related to what's being said in the video. Okay, so once you've watched that, then you want to go download this Beetroot Lab. Okay, so to download, you can just scroll down, and then there is the download button. Okay, so you download it, and actually I will download it, and then you will open it, and what you can do is you will be able to type the answers directly into the document. Okay, so what is the purpose of this experiment? Has anybody looked at the experiment yet? Do you know what the purpose of the experiment is? Anybody want to tell me? We have how many people here? 7 people here. Did any of you look at the lab by any chance? No. Okay. So you would watch the video, and then you would think about what the purpose is, and then you will say you will type the answer here. You can obviously like delete this thing, and then you say the purpose of this experiment is to investigate, and then you figure out what it is. Okay, so you will do this, and then they will tell you how many marks each question is. You write a hypothesis. Remember, the hypothesis doesn't have to be correct, but you do have to write it, in full sentences, right? And then so on and so forth. Okay, so you will finish this whole lab. You will have to do a graph, right? Here it says below are some spectrophotometry data, and then you can use Excel or Google Sheet, or whatever graphing software you have to do a graph. If you don't know how to do a graph, you can watch the YouTube link to see how to do it. But I believe you learn how to do graphs on Excel from your computer class, right? Is that correct? Can someone confirm that for me? Not yet, or yeah. Oh yes, okay, good. Yeah, so that's no problem. But I mean, if you're super stuck and then you don't know what to do, don't know how to do a graph, you can send me an email, and then we can set up a Zoom meeting, and I'll show you how to do it. Okay, but that's like 4 marks. Make sure the graph is labeled properly, right? In the past, like, you wouldn't believe me, like some people would just like, I'm not making this up, like someone would just like do this, right, and then take a picture and then put it on. Like, how am I supposed to give you marks for something like this? Okay, this happened like last semester, more than once too. So make sure you do it nicely, and then you will get your grades. And then there is some analysis. It's very important that you try to come up with the answers yourself. Don't go scouring on the internet and try to find the answers. Okay, because what you find on the internet might not apply to what we are doing. Okay, sometimes it sounds like it's the same experiment, but not necessarily. Okay, and you know, I've marked enough of these papers to know when you are, you know, answering the question yourself, or if you are just, you know, copying from the internet. Okay? I must have marked like over, I don't know, thousands of these labs over the course of my teaching time here. So, you know, make sure you try to use your own words, okay? And you know, just justify your answer, right? And there's no need to, you know, find the answers on the internet. So afterwards, right? Yeah, you will save. I'm not going to save here, but you will save, and then you will go submit it. Okay, so I'm going to go back to the home page, and then here is the assignment folder. Okay, so you just click into it, and the assignment is not available. Okay, I'll fix that. Okay, I don't know why it's not working yet, but I'll fix it after class. Okay, and so when you go into it, you will see the two assignment folders. Okay. Oh, it's not available yet because it's you know, it's available in August 8th. Right. It will be available by Monday because I assume you won't be done on the weekend. But it's okay. I'll open it after class, and then you will click into it, and then you will submit it. Okay. Let me see if I can change that right now. Sorry, I seem a little bit incoherent today. Let me go here. Edit the folder and submission availability. I will make it available like right now. You know, let's make it available yesterday. Save and close. Okay, so now I go back to as a student. You go submit it in the assignment folder. So you click in the assignment folder, and you will see that you can submit it. Okay, so you scroll all the way to the bottom. You say add a file, and then you know, from my computer, and then you know, wherever you save your file, whether on the desktop or in the folder, you just drag it in, and then you say add. Do you guys understand? Okay, so for example, like this is the one that I have, the Beetroot Lab, the one with the answer. You drag it in like that, and then you click add. Okay, let's do that and see what happens. Now, so there it is. And then if you have something you want to tell me, you can tell me. Okay, I don't know. Some people say that, you know, they have some concerns about whatever. Doesn't matter. If you want to type a comment, you type a comment. If you don't want to, that's fine as well. Okay, and then you click submit. Okay, but the upload button is all the way at the bottom. Okay, this is just a marking rubric, right, for me to grade your stuff easily. Okay, so there's no need to pay too much attention to that because it's just the score for each of the questions. Okay, and then you click submit, and I think you get an email confirmation, right? Okay. So I won't mark it until the deadline. The deadline is Friday, October 8th, okay, at 9:10 p.m. And you know, if you change your mind throughout the week and you want to submit another one, you could do it. But the system will only keep the most recent one. Okay, so it will override your previous one. Sometimes people, you know, revisit their assignment and find a mistake, and then they go change it. Okay, but try to hand it in before Friday. Okay, so you know it's done. You get it over with. Any question about the lab? I know some of you are just joining. I was just explaining how to do the lab and submit it. Anybody have any comments, concerns? It's pretty straightforward, right? Okay. Now but seriously, take the time to do the lab. Do it well. It's an easy way to get 10%. But in the past, people just, I don't know, I don't understand. Like, I guess people procrastinate and they don't do it until like the day of, and then they just hand in these really, really terrible answers. Okay. And you just give away 10%. We don't want that to happen. Okay. It's not ideal. Okay, and then afterwards, you will have the engagement week. You do absolutely nothing related to the course, and then we will do the lecture 5 when we come back. All right. So I hope that makes sense. Now let's go back to this thing. Complete the following diagram to illustrate the relationship between anabolism and catabolism. Okay, so in general, let's quickly define what they are. Anabolism are basically reactions to build macromolecules. Okay, macromolecules. Whereas catabolism or catabolic reactions, they are reactions to break down macromolecules. Okay, together they make up your metabolism. Okay, so here is a diagram. Over here, all these things, these are macromolecules from our food source, right? Macromolecules from food sources. Okay, so like you eat a pizza, then you would have some proteins from the cheese, you would have some lipids from the olive oil and the cheese, and you would have carbohydrates, right? From the dough, okay? And all these macromolecules we would need to break down, okay? So when we break down these macromolecules into their monomers, you would extract the energy, okay? And then the energy will be captured to produce ATP, okay? Adenosine triphosphate, right? This is the preferred form of energy in the body. And it's not a very efficient process, right? Not 100% of the energy from the food will be captured in ATP, right? A lot of it is actually lost as heat. Okay, I think we're just about like 40% efficient. And the rest of the energy is wasted as heat. And when I say wasted, it's not completely wasted, right? Like we need to keep our body temperature at 37 degrees Celsius, right? So a lot of that heat is used for just that purpose. Okay, so this way, right, breaking down the macromolecules into the building blocks, this is the catabolic reaction. Catabolic reaction. And then we will use the building blocks to make things that we need. Okay, maybe we need to build a hormone. Maybe we need to, build phospholipids. Okay, maybe we need to store the extra energy as glycogen. So all that building, it's going to be the anabolic reaction, okay, or anabolism, okay? And this is going to require energy. When you build things, it's going to require energy. So you will use the ATP generated from the catabolic reactions to make the molecules that we need. Okay, so these are macromolecules needed in the body. So essentially, this is what happened. This is the takeaway message. Okay. Energy released from catabolism is used to drive anabolism. Okay, that's why we have to keep on eating. Okay, we need to eat to keep ourselves warm, and we need the energy from the food that we eat to build things that we need. Okay? Questions, anyone? Okay, so let me quickly explain how ATP works. Okay, so ATP is, as I said, adenosine triphosphate. All right. Oh, Shannon asked, where does the heat come from? So the heat is basically wasted energy, right? As I said, we are not efficient at converting the energy. So a lot of the energy is lost as heat. So maybe let me explain what efficiency means, okay? Efficient. Okay. Does anybody still have these light bulbs at home? Like the really traditional one that you screw on, as opposed to, you know, the newer one that looks like this. Okay. What are these light bulbs called? Anybody know? These are fluorescent, right? Or LED even. Okay. The older ones, yeah, exactly, right? The incandescent, right? So if you turn on an incandescent light bulb for, I don't know, even 10 minutes and you touch it, what's happening, right? It's hot, right? So the incandescent light bulbs are actually very inefficient. What's the purpose of a light bulb? The purpose of a light bulb is to convert electricity into light. If it's efficient in doing that, you should not be generating a lot of heat, which is why you touch these fluorescent, it's not very hot. And if you touch the LED, it's virtually not hot at all. So they are getting more efficient, right? They don't waste energy, okay? Compared to like a light bulb, which is only like less than 5% efficient. So we as a human being, right? We would like to convert all that food energy into energy we can use, ATP. But during the conversion, right, we waste a lot of energy as heat, just like the incandescent light bulb. Does that make sense? Okay. So adenosine triphosphate, right? So we have the adenosine, okay, and then which is connected to sugar, but I'm not going to draw it. And then you're going to have like, you know, P, and then a special bond and a P. Okay, so this last bond here that connects the second P to the last P, the last phosphate, this is a very energetic bond. Okay, so this here is ATP triphosphate. And what happens is you can break it down. You can break this down into 2 phosphate plus 1 phosphate. So this is ADP, adenosine diphosphate. Okay, and in order to do that, you have to like add water to it. Hydrolysis, right? And what this does is when you break off that last P, you will be releasing a tremendous amount of energy. Maybe not tremendous, we'll just say it will release energy. Okay, don't make it sound too dramatic. And that energy is used for various things, one of which is to drive the anabolic reactions. Okay, so let me write here, energy used for anabolism and other things. Okay, and then what do we do with the ADP and the P? Well, we have to recharge it. So recharge it. Okay, so we need to supply energy to recharge this thing. Okay, energy has to come. What is going on with my pen here? Okay, energy has to be inserted into the ADP to charge it back to ATP. So this is energy from, you've guessed it, catabolism. Okay, and then this just keeps on going, cycling between ATP and ADP, ATP and ADP, as long as we are alive, basically. Okay. Are we good? Any question? I know I keep on asking you like every 5 minutes, but you guys very quiet, so I just want to make sure that we are okay. All right. What else do we use the ATP for? Well, we can also use the ATP for things like mechanical work. Mechanical work. So an example of mechanical work would be like movements of cilia, right? Or flagellum. that requires energy. And you will supply it in the form of ATP. Another type of work that you can do is chemical work. Chemical work. Okay. So, you know, many reactions requires the input of ATP, just like I told you, right? Such as anabolism. Okay. Even sometimes if you need like enzymatic activity, that requires ATP. And finally, transport work. Transport work, right? So like active transport. Right? If you want to move things from low concentration to high concentration, that is transport work that requires ATP. Okay. All right, let's do some polls. Let's do some polls. See if you guys understand what's going on. Go for it. Okay, only 5 responses. I'll wait for a little bit longer. Okay. All right, let's take a look at this one. Okay. Catabolic reactions, right? So catabolic reactions are going from polymer, right? The macromolecules to the monomers, right? Okay. So in doing so, it's going to release ATP plus heat. It's like the stuff on the left side of a diagram, right? Okay. So consume more energy than they produce. That is not true because you will be producing energy and we will use the energy to make ATP, right? Breaks down polymer into monomer. That is correct. Okay. Produce excess heat to warm the body. That is also correct. I think I launched the wrong poll. I should have launched like the multi-select poll. Sorry about that. Not at my greatest performance this morning. Only occurs in human cells and not others. So that's not true. Okay, all cells would have some kind of catabolic pathway, and so you know these two would be the correct answers. Okay, let's try the next one. Oh, this one we have to talk about first before we do it. We'll come back. We'll come back to it. Okay. Let's see. All right. List 4 factors that affects the rate of diffusion. Anybody help me out here? Give me 4 factors that affects the rate of diffusion. Does anybody know? Yeah, very good. Marisha said size, temperature. Okay. Yeah, mass is size and mass. Similar concept here. Steepness. steepness of concentration gradient and charge of molecule. Charge, we'll just say charge. Okay. So generally, smaller equals faster rate. Okay, which makes sense. If it's smaller, it's easier to move around quickly, right? For temperature, higher temperature equals faster rate. Okay. And steepness of concentration, what does it mean? It means between the 2 points, are the difference big difference or is it small difference? You can think of it as like a ball rolling down the ramp. Okay. So like if you put a ball here, it goes down, right? But if the ramp is like steeper, then the ball goes down faster, right? Okay. So similarly, if I have two chambers, okay, and then this here is like, 70% salt, and over here is 30% salt. So the difference is 40% compared to, let's say we have 70% on the left and 60% on the right. This would have faster diffusion. Okay, and this would have slower diffusion initially, okay? Because the concentration gradient, the difference between the 2 points is bigger. Okay, gradient just means difference, right? Okay, and then the charge, right? So let me explain the charge. Let's say you have a chamber, right? Membrane, and then you have a bunch of H plus here. And this is something we'll talk about in lecture 5, okay? And then you have like maybe 1 H plus here, okay? 2 H plus. So there are two driving forces that will push the H plus over. Number one, we have the concentration gradient, right? You have high concentration of hydrogen ion on one side and low concentration on the other. So it will, naturally move from high to low. And secondly, we have what we call like an electrostatic gradient. Okay. Like charges repel each other. So if you have a bunch of positive charges very close to each other, they would push each other away. And that creates a force that would help move the H plus from one side to the other. Okay. But here's the thing, right? As time goes on, as the H plus gets pushed towards the bottom here, then you would build up a positive charge here as well. So eventually, the electrostatic gradient would slow down. Okay, because now all the positive that went to the other side is going to try to push back up, right? So eventually you will reach what we call an equilibrium, where the two sides are equal, and then the movement would stop, and no changes would occur afterwards. But these are the 4 factors that would affect the rate of diffusion. Okay. And then we talk about what happens when you put a cell in various types of solutions. Okay, so let's start with the first one, right? Isotonic solution. Isotonic. What does isotonic solution mean? Well, isotonic basically means that the concentration of the solute, okay, basically everything that's dissolved in the solution, is the same on the inside of the cell as it is on the outside. Okay, so we say the solute concentration. Can I just do this for concentration, short form, right? The solute concentration is the same on the outside and the inside of the cell. Okay, the environment has the same concentration as the inside. Okay, so like when we do IV into our body, we want to make sure that the solution is isotonic, right? So that our cell is not gonna shrink or burst from it. It's in rare situations. I think I mentioned this in the lecture. Sometimes we would do hypertonic or hypotonic. right? If the person blood is too concentrated, right? Because of dehydration, for example, then you might want to give them a little bit of hypotonic solution to help dilute the blood a little bit, okay? But generally speaking, if you just do IV, we want it to be isotonic, okay? So what happens is the movement, the rate of water entering the cell is the same as the rate of water leaving the cell. So there is no net water movement. Okay. Cell is normal. Okay. Now, one confusion is that people think the water does not move at all. That's not the case. Okay. It just means that, you know, whatever amount of water is coming in, that is the exact amount that's leaving at the same time. Okay. So overall, they cancel each other, and the cell is not going to change shape. So that's the normal condition for a cell. This is a red cell, right, in our solution. Okay, next one. Next one is hypotonic. Hypotonic. What's going on with hypotonic? Okay, so solute concentration outside is less than solute concentration inside of the cell. Okay, so you would have more dots on the inside. You can't really see it in this diagram, I guess, but trust me, it is there. Okay, you can add it in and make it look like a poppy seed donut. There you go. There you go. So you will have more water coming in than leaving. Okay. So watering, rate of water entering is greater than rate of water leaving. Okay. Then what happens is net water movement equals to into the cell. Cell will swell and potentially burst. Okay, we call this lysis. Okay. Or we say the cell lyse. Okay. The cell lyse. Okay. But as a noun, this is called lysis. Okay. Next one, hypertonic. Okay, hypertonic. So that is when the solute concentration outside is greater than solute concentration inside. Okay. So rate of water leaving, or let's just keep it consistent, entering will be less than rate of water leaving. Okay, so you have more water leaving the cell than coming in. Okay. Leaving, okay, leaving, and then you have some coming in. And again, right, there's much more dots on the outside. Than inside. Okay, so net water movement, net water movement is out of the cell. the cell will shrink. Okay, and this is called plasmolysis. Okay, plasmolysis. Okay. Yeah, this is quite an important concept. Now, sometimes you will see a diagram like this. Okay. I don't remember if I have a diagram like this in the lecture, but you would have a U-tube, a U-shaped tube. And then there's a membrane here. Okay, only permeable to water. Only water can pass through. Okay, so you would have some water here. Water. Okay, and then you would dissolve some salt here, salt or other solute. Okay, and then here. And if you let it sit, what's going to happen to the water level on each side? Okay, what's going to happen to the water level? So let's think about it. A, no change. B, left side drops, and right side rises. Okay. C, left side rises, and then right side drops. Okay. Let's do a poll. My question is, if I leave this alone, what will happen to the water level on both sides? No change, or is it B or is it C? Let's see what you think. Okay. Now, only 5 of you answered. I'm not sure where the rest of you are on this position. And a lot of you choose B, saying the left side will drop and then the right side will rise. Can someone explain why that is your choice? I would like to hear the explanation before I explain it. Yeah, you can type it or you can unmute is probably better if you want to explain it. Why the 4 of you who chose B, can you explain to me why that is the case? Why would the left side drop? To equal out the solute concentration. Okay. The solute are the red dots, right? And these are the solutes, right? Okay. Can the red dots move? That's a question I want to ask you guys. Will the red dots move from one side to another? No, right? Because the membrane only allows water to move. Okay. So the question you want to ask yourself is, which way will water move to equal out the concentration? Right? You have more solute on the left side, less solute on the right side. Which way will water move to make them the same? Right? If water moves to the right, let's see what happens. Think about this. If water moves to the right, you will make the right side even more dilute and concentrate the left even more, right? Okay, so that's not what's going to happen. To even out the concentration, the water will have to move to the left. Right? So as water moves to the left, this is going to drop down, and then this is going to rise up. Okay, and when that happens, right, the left side will be less concentrated, and then the right side will be more concentrated. Okay, so the correct answer is actually C. Actually C. Does that make sense? Anybody have any question about what this means? Clarification? Sure. All right. Let's take a break. Okay, we'll continue when we come back. 5-minute break, so we'll come back at like 9:41. All right. Hold on. There is a question. I'm just gonna. It's a private message, so I'm just gonna read it. So it's not about the charges, but more on the side of its concentration. Not quite sure what that means. None of these things are charged though. Okay, it's just like salt, right? As I mentioned. And I think the general rule is this. Okay, like if you have to put like a summary line to this. Okay, this is the key thing to remember. This is the key thing to remember. Water will always move towards the more concentrated side. Okay. And this is the more concentrated side, so water is going to move that way to try to dilute it out. Okay. Hope that makes sense. Yeah, that's okay. If you have to go, you can go, and you guys can watch the recordings after. Okay. All right, let's take a break. Let's go 5 minutes, and we'll resume at 9:42. Hello again. Let's keep going. All right, next we have compare passive transport and active transport. Okay. Let's start with the left side. Active transport. So in active transport, the movement of molecules go from low concentration to high concentration. So this is against the concentration gradient. On the other hand, the passive transport, it is going from high concentration to low concentration. Right? So this is called down its concentration gradient. Okay? Down the concentration gradient. Okay? And over here, this is against, right? Sorry, against concentration gradient. Now, because you're going against the concentration gradient, you're going uphill, right? Imagine pushing a big, I don't know, boulder up the hill. That's going to require energy. So yes, okay, requires energy from the cell. Okay. And on the other hand, passive transport does not require energy from the cell. Okay. Example of active transport would be things like, assuming you've watched the lecture, there will be like endocytosis. Okay, so like eating a cell, right? We talked about it last time, right? The phagocytosis. Another example of active transport would be things like a protein pump. Okay, so these are like the sodium potassium pump, right? That's an example of a protein pump. Passive transport would be diffusion, osmosis, and facilitated diffusion. These are some examples. Okay. Let's do another poll. We just talked about it, so you should be able to find the answer. Go ahead, try it. Okay, so only 3 of you answered. The correct answer is B. Okay, D is not correct because movement of water from low concentration to high concentration, that's just osmosis, right? Okay, and that happens on its own. Okay, next one. Oh, check all correct answers. Sorry, I launched the wrong poll again. Go ahead. Just curious, like some of you are not participating. Is it because you cannot see the question? You cannot see the poll on your device, or like you need more time? What is it? So that I could help you with it. Or maybe you're not there. Not quite sure. Okay, so we have answers all over the place. Let's see. This is a hypertonic solution. Right? Hypertonic means the cell is placed in a solution that has more solute outside. Okay, so net movement of water will be out of the cell. The cell will shrink plasmolysis. That's the term for it. And then the solute concentration is higher outside compared to inside. The answer is A, C, and E. Okay. Yeah, we tried this one as well. So remember, the membrane is phospholipid bilayer, and there is a thick hydrophobic layer in between. Right? So anything that's hydrophilic will have a hard time crossing it. Right? So it's either B or C, right? Hydrophobic, right? And of course, if it's smaller, it will be easier to cross it. So the correct answer is C. Okay. If it's charged, it won't be able to cross it easily because charged particles don't interact well with hydrophobic substances, and the membrane has a thick hydrophobic middle layer. All right, let's see. Okay, this one is very, very hardcore. You can try to do that. Maybe maybe you guys don't feel comfortable sharing in a big group. I am gonna try this breakout room function. I'll put you guys into small groups, and then maybe you guys can unmute and talk about it, and then we'll come back in about 2 minutes, okay? And then we'll see if you have better answers. Just try it out, okay? See how it goes. All right. So it's gonna create like a invitation to go to a breakout room. You just say join, okay? So go ahead and So how was that? Like, did people talk, or was it just awkward silence? I want to find a method that we can all participate in. So I see some of you came back. Was there a discussion going on in the room, or you feel like probably best not to do this again? Just give me some feedback, okay? So I know what works for you guys and what does not. The time was not too short. So you guys were discussing in the room. Was that correct? How about the other groups? Were people discussing in the discussing about the question? Obviously, we could make that time longer next time if there were actual discussions. What about the other two groups? There were three groups. Each group had two people. We you only said hi. Okay, so there's was not much discussion. Okay. What about the other groups? Can you let me know? Like just type something so I know whether this was a helpful thing to do or not. You needed the question posted. Oh, so when you go into the room, you cannot see the question? Is that the problem? Oh, I did not know that. Oh, okay. So sorry about that. Okay. I will figure out how to make that happen next time. Okay. That's an oversee. That's an oversight I have. Okay, so I'll make that happen next time. Thank you for the feedback. We will fix that. Okay, so do you guys have access to the chat box while you were in the room? I haven't done bigger room before, so I'm not sure. Like, are you able to see the chat box while you were in the bigger room, or did people not? Yeah. Okay, so I'll put the question in the chat box next time, and then we will see what happens. Anyway, let's take up this question. So he has a, and people who are watching this on YouTube, you probably skipped all that, right? So we will put a bag here, and the bag has salt water solution. Okay, and then this is pure water on the outside. What's gonna happen? Well, what's gonna happen is this is actually a hypotonic solution, right? So water will go in. Okay? So the volume of the bag will decrease over time. Nope, it will increase. pH will change. Nope, because we are not changing the pH. pH is a hydrogen ion, as we learned earlier. So there's no movement of hydrogen ion, no change in pH. This solution, the flow of water into the bag will never equal to the flow of water out of the bag. Maybe. Let's move on and see if there are other examples. The net flow of water will be from the bag to the surrounding water. That's not true. It's the other way around. And the net flow of water will be from the beaker into the bag, causing the contents of the bag to increase, right? So E is the best answer. Okay, E is the best answer. Is that okay? All right, let's do one more. Okay. You know what? Let's try the breakout room thing again and see if it's gonna work. Okay, so this is the question in the chat box, and then these are the answers. Okay, A, B, C, D. You can fill that in again. Let's try the breakout room one more time, and then you guys give me some feedback. I'll give you like maybe 3 minutes, 2 minutes to think about it. Okay, so breakout room, go. Okay, thank you for doing that. How about this time? Were there productive discussion? Do you all have a chance to talk about it this time, or what? Again, give me some feedback so I know whether to do this or not, or you just prefer the poll without the room. The chat box disappeared. Oh no! Oh my goodness, this is terrible. Okay, so we'll have to figure out a way to do this in the future. You spoke. Okay, good. I think we need a little bit to think over answer. Okay, so is this something that you all of you prefer to try again? Maybe like this time with the questions, right? Like maybe I'll post the question, on a document before class, and then you will have access to it afterwards. Okay, maybe we can answer questions. Study guide. Yeah. Okay. So people are open to this, right? Right. We can do this because it's a small class, right? Okay. Okay, thank you for the feedback. I just want you guys to have a way to, participate, right? Because otherwise it's very boring for you, I suppose. Like, I mean, I sit in Zoom meeting like for the school, and sometimes I doze off after hearing the speaker for like, an hour, and you have to listen to me for 2 hours. So I just want ways for you guys to interact. In any case, what is the answer for this one? What discussion, what conclusion did you guys get? Okay, so for those of you who participated, you all make the choices. Okay, well, actually, the answer is C. Okay, maybe it surprised some of you that the answer is C. That's because, and listen carefully, this is going to help you with one of the lab questions. The size will not change because if you freeze the animal cell, you guys ever put a can of Coke in the freezer before? Anybody did that before? Or like you fill up your water bottle to the brim, you close the cap and you put it in the freezer. Anybody did that before? What happens when you try to take it out after leaving it overnight? Any experience of that? No? Am I the only one who has put a can of Coke in the freezer before? If you do that, it actually might burst open. Okay, that's because as water freezes, it expands, right? As water freezes, it expands. We know that. You can try it with just water bottle. You can see the ice expanding. So what happens is when you freeze an animal cell, the water in the cytoplasm will expand, and that actually will destroy the cell membrane. Okay, the ice crystal will actually pierce the cell membrane, make holes in it. Okay, and afterwards, when you go back to room temperature, the cell membrane is full of holes. So it doesn't matter if you put it at hypertonic or hypotonic, things will just go in and out without any regulation. And as such, the animal cell will not change the size. Does that make sense? I hope it makes sense. Okay, so let's go back to the study guide here. Choose the more concentrated solution in each of the following solution. Okay, so over here we have 80% water and 20% starch versus 90% water, 10% starch. So more concentrated means more solute. So higher starch percentage means more concentrated. Next one is maybe a little bit more complicated because you have to do a little bit of math. 5 grams of sodium chloride in 50 grams of water versus 5 grams of sodium chloride in 25 grams of water. Okay, perhaps the easiest way to do it is to change the water to the same amount. So, how do I get from 25 to 50? I would multiply by 2, right? So 5 grams in 25 grams of water is the same as saying 10 grams of sodium chloride in 50 grams of water. Does that make sense? Because I multiply 25 by 2, then I have to multiply the NaCl by 2, right? It is like ratio, right? So now I can compare them because it's the same amount of water. So 5 grams in 50 versus 10 grams in 50. This is the more concentrated one. Okay. 85% solvent, 15% solute versus 75% solvent and 25% solute. Okay, so more solute means more concentrated. Next, 3 types of bulk transport. We have phagocytosis, which actually is a type of endocytosis, right? We have pinocytosis. Right? We also have like exocytosis. Right? And then there is another one called receptor mediated endocytosis. Okay, so these are different types of bulk transport. Cell eating, like eating a bacteria. Cell drinking, right, like bringing in large amount of extracellular fluid into the cell. Right? And this one would be triggered by the binding to a surface protein. And then exocytosis would be releasing material into the surroundings. Okay, next one. Long answer question. Any requests here? Do you want me to take up any of these particular ones? Like comparing contrast of these one, is there a particular one that you are not familiar with and you want me to talk about? Please let me know. The sodium potassium pump. Okay, no problem. Let's do that. I'll do it down here. I will go all the way to the end. Okay, so sodium potassium pump. I think I talked about that in the lecture, but here we go again. So this is the cell membrane. Cell membrane. Right, this is the outside. This is the inside. And you would have the sodium potassium pump like here. It's an integral protein. Sodium potassium pump. So the key things you have to know for the sodium potassium pump is it will pump 3 sodium out of the cell in exchange for 2 potassium. 2 potassium. Okay, so over time, as this is happening, right, you will have high sodium concentration on the outside and low concentration of potassium on the outside, because you keep on bringing the potassium in and you keep on bringing the sodium out, right? And then on the inside, you would have low sodium because you keep on pumping them out, and you would have high potassium. Okay. So as you keep on doing this, you can see the sodium goes from low concentration to high concentration. And same story for the potassium. It's going from low concentration to high concentration. So this is going against their concentration gradient, which is why this process will need ATP. Okay. That's basically the key idea of sodium potassium pump. As to why we are doing this, why are we pumping sodium out and potassium in? It turns out your neurons relies on this sodium potassium pump to function, and you will learn all about it next semester. Okay, but that's basically what you have to know for sodium potassium pump. Any follow-up questions, or is that adequate? Okay, good. All right, what about comparing similarity and differences? All good for these? Okay. Application questions. Let's quickly talk about them. In 2007, a woman who competed in a radio station contest to see how much water she could drink without going to the bathroom and died of water intoxication. Okay. Explain why drinking a large amount of water in a short period of time could result in water intoxication. So basically, if you drink a lot of water, just like tap water, then you are diluting your blood, right? Okay, so you're diluting the blood, and creating a hypotonic environment. Okay, and this will cause the water to go into the cell, and a lot of the cell will burst, right? Particularly your red blood cell, RBC burst. Okay, and with a lot of RBC bursting, you will have what's called like hemolysis. Right? Hemo is like red blood cell. Lysis, it's breaking, and then you won't be able to transport oxygen. Right? So that's one reason you might die. A second reason you might die is you would have a very low electrolyte. Okay, so electrolytes are like things like calcium, magnesium, right? Sodium, all these things have functions in the body. You need calcium for contraction. So if you dilute all those calcium, then your muscle might not be able to contract, right? Your heart is a muscle, so that could kill you. Okay, without the calcium, you also might have a difficulty for the nerves to communicate, and that could lead to a host of other problems as well. There's a question. So is it the same as not quite the same as drowning? Because drowning is the water going into your lungs and you cannot do gas exchange, you cannot breathe. This is, you can still breathe, but then everything in the blood is diluted, and they're not going to be able to perform at a level at the right concentration, and that's what's causing the problem. And also your red cells are going to be bursting. All right. So if someone is suspected of water intoxication and they're not dead yet, this would be a good time to give them a hypertonic IV, right? Okay, to kind of rebalance the concentration. Does that make sense? Next one, single cellular organism that lives in fresh water are constantly exposed to hypotonic solutions. Speculate what kind of transport mechanism could be used to prevent lysis. So, last time we talked about this thing, right? This is the paramecium. I don't know if you remember. It's like a protist that lives in pond water. And pond water is hypotonic, as opposed to like seawater, which is hypertonic, right? And so water will come in all the time, right, at a greater rate than it's leaving. So these organisms usually would have some kind of contractile vacuole. Okay, so there's a special vacuole in them. Okay, and there is like muscle around it, like microscopic muscle. So it will suck up the water in the cytoplasm, and then it will push the water out. Okay, so that's called like a contractile vacuole. Okay, and that's how they prevent themselves from bursting. Okay, and interestingly, for what do you call that? The marine organisms, like fish that lives in the ocean, they would have to do the opposite. They would have to conserve as much water as possible without losing it to the environment, right? Because then it's a hypertonic environment all the time, and you are at risk of dehydrating yourself if you don't have mechanisms to counteract that. Okay, and some of it's done by having special structure in the kidneys, for example, and you will learn all about it again next semester. Finally, salting has long been used as a method to preserving food. Coating meat with a layer of salt will dehydrate the meat as well as any microorganisms. Explain how this process works. It's basically what I just said, right? The salt, if you put a lot of salt around the food, it sucks the moisture out of the cell as well as the microorganism, because it's a super hypertonic environment, right? And by sucking out all the water, right? The microorganism would dry out and die. And without microorganism, it is not gonna spoil the food, and you can preserve it for much longer. But preserved food is, of course, not too healthy to eat. For a whole set of other reasons, right? Consuming too much salt is not good. And a lot of preserved food nowadays, instead of just using regular NaCl, there are other preservatives that is being used, right? Nitrates, the sodium benzoate, all those things are associated with a higher risk of developing colon cancer, right? So might not be a good thing to eat too much of those things. Okay, fill in the blanks. The plasma membrane contains 2 layers of phospholipid. Something are important for maintaining the proper fluidity of the plasma membrane. That is cholesterol. Maintaining. Okay, cholesterol. You might have heard that too much cholesterol is bad, but those are like cholesterol in the bloodstream. But we also need cholesterol in the cell membrane. By having more cholesterol, it increases the fluidity, so it's not going to dry up and it become too solid. But you don't want too much either. Too much is going to make it too leaky. Okay, so you need to have to just the right amount to have the right consistency. Breaking down molecules into smaller unit, that would be catabolism. And then the opposite of catabolism is anabolism. Okay, catabolism, kata is the same root word as catastrophe. And catastrophe, everything breaks down. So catabolism, you break things down. That's how you can remember it if you're getting confused. An ATP molecule contains adenosine, sugar, and 3-phosphate group. Osmosis is the diffusion of water. Carrier protein and channel proteins are examples of facilitated diffusion. So these are diffusion that involves the help of a protein, of an integral protein usually, and it does not require energy. Okay, so no ATP requirement, no ATP needed. An animal cell will shrink when it is placed in a hypertonic solution. And then a hypotonic solution has lower solid concentration than the inside of the cell. So all the water comes in and the cell will burst. Adenine and adenosine is not exactly the same thing. I think adenosine is like oh, okay, I see what you mean here. Yeah, here I should put like adenine. So adenine is like the actual molecule. That's what it's called. And then when it's connected to the sugar and then the phosphate, then we call it like adenosine. Okay, so maybe we stick with adenine. Thank you for correcting me. Cell eating is called phagocytosis, and cell drinking is pinocytosis. A cell undergoes lysis when placed in a, oh, sorry, this is hypertonic, so it's going to undergo plasmolysis. All right, so the quiz next week is both lecture 3, which is the cell organelle stuff, as well as what we learned today. We still have some time, so we can do a Kahoot if you want. Let me turn off my camera, and we can do this. I don't know if you played this in your other courses before, but you would go to this website. You can key it in your phone, in your browser, www.kahoot.it. And when you're there, you can key in the game pin. Okay. And then we will do 10 practice questions for the quiz. I will type the website in the chat room. Anybody having trouble joining? We have 3 people now. Oh, we lost one. Okay, well, it doesn't look like there is a lot of interest in this. Okay, we have another one now. Okay, we will start with 4 people, I guess. It's rather sad, but we'll go. Thank you for joining. If you're still trying to join, the game pin is at the bottom right. Rough ER, right? That's the organelle that forms a continuous channel with the nuclear envelope, right? Remember, the nuclear envelope is the membrane that goes around the nucleus, and then it will continue to extend into the cytoplasm to form the rough ER. Oh, you're doing it on the phone? Oh, sorry about that. Yeah, that's okay. You can still do it. You can still join if you want. There's the pin code, or you can just do the question yourself. That's okay as well. Okay. The smooth ER is where you produce lipids, right? Remember? Okay. The ribosome, which is what some people choose, the ribosome is for protein synthesis. Okay. And phospholipids and cholesterol are not proteins, right? The mitochondria is for energy production. So the correct answer is smooth ER. That's okay. Now you know what you need to work on. Let's try number 3. Okay. Detoxification. We talked about this in the lecture last time in the tutorial actually. Smooth ER, in addition to making lipids, it would also help break down poison and drugs. Okay, lysosome is more for breaking down things that have been ingested by the cell, like a bacterium. or break down all organelles, as well as like, carbohydrates and lipids, that sort of stuff, okay? So smooth ER is to break down toxic material from like drugs and poisons, okay? Okay, we learned about this today, right? It recognizes the foreign antigens, right? The foreign proteins on the invading cell. That is an example of cell-cell recognition. It's not talking about eating them, right? If it says white cells engulf an invading cell, that would be phagocytosis. This is talking about recognizing the invading cell. That would be an example of cell-cell recognition. Yep, hypotonic, it will burst. Okay, it will burst. Hypertonic, it will shrink. Phagocytosis requires the use of ATP, right? Because it's an example of bulk transport. Facilitated diffusion, as I mentioned, in the fill in the blank, like just now, right? You see, carrier proteins and channel proteins are example of facilitated diffusion. No ATP is needed because all that means is the molecule cannot cross on its own, so it's going to rely on the help of carrier protein and channel protein. It just provides like a tunnel for them to go through. You don't actually need to have ATP. Okay, so the correct answer is phagocytosis. Osmosis is just diffusion of water. It will happen on its own. All right. Does not affect the membrane permeability. Okay, so we talk about temperature, right? Temperature will affect it because if it's very hot, then the membrane becomes more leaky, right? Because the molecules are further apart. pH is going to affect it because when you change the pH, we talk about how pH can affect the shape of the protein, right? That was like the lecture number 2, right? By changing the pH, the sum of the proteins is going to unfold, and remember there are a lot of integral proteins in the membrane. So if you unfold some of those proteins, it is going to cause the membrane integrity to fall apart. Membrane composition will affect it because if you have more cholesterol, for example, it will be more leaky, right? And so the size of the cell does not matter. Okay, you can have a big cell, you can have a small cell. If both of the cell is made up of the same same molecules for their membrane, then they will have the same permeability. Okay, so the size of the cell does not affect membrane permeability. You should keep this in mind when you do the lab. Okay, and if you missed what I said, just watch the recordings. And a couple more questions. The mitochondria has a double membrane. Remember, we labeled it. There's an outer membrane and there's inner membrane that folds to form the cristae. All the other ones only have a single membrane. Okay. The ribosome has two subunits, right? Like a big one and a small one. Maybe that's why people are confused, but it doesn't even have a membrane, the ribosome. And lysosome only has one membrane after it pinches off from the Golgi apparatus. Okay. In the lecture, I said if compared to an animal cells, then yes, you will only have chloroplasts in plant cells, but not in animal cells. However, I also did say last week that chloroplasts are found in bacteria sometimes. They are found in protists. Okay, so they're not exclusive to plant cells. But I think what all of you were thinking is that yeah, it's in plant cells, but not animal cells. But that's not what the question says, right? It says only found in plant cells. And that's not true because, like I said, it can be found in bacterium or sometimes protists. Okay, last question. No centrioles, that's correct. Centrioles are the little T-shaped organelles, usually comes in a pair, and they produce the spindle fibers that help guide the chromosome movement during cell division, and plant cells don't have that. Okay, so that's the end of the review questions. Now, I am a little bit concerned. Well, only 4 of you participate, so I don't know, if the rest of you are getting the answers. There's no way for me to tell. But judging from this, looks like there are some studying that needs to happen for Tuesday's test, so quiz. So make sure you do that. And if you need help, okay, you need to find me for extra help, okay? Like I cannot help you if I don't know what the questions are, right? What your concerns are, right? So if there are some concepts that you don't understand, just email me, or if you need to have a one-on-one chat with me, we can arrange that as well. All right. So best of luck next week. And again, no tutorial next week. And then afterwards is engagement week. So that means I won't see you until 2 weeks later. All right. But I'll be available on email for sure. Okay, that's it. Thanks again. And I'll see you next time. Bye for now.